Good morning, everyone. I would like to thank the organizer for the invite to participate in this forum. My name is Sowemimo from Obafemi Awolowo University, Nigeria, and I will be presenting on the topic, current trend of the prevalence of Toxocariasis in humans in Nigeria. Next slide, please. What is Toxocariasis? Toxocariasis is one of the most widespread zoonotic parasitic infections and is listed as one of the 5 most important neglected tropical diseases by the Center for Disease Control USA. Toxocariasis is caused by the larvae of Toxocara canis or Toxocara cati, which are intestinal ascariid nematodes of dogs and cats, respectively. Next slide, please. Toxocara eggs are usually passed out with the host feces, and when it gets to the environment, it becomes mature under suitable environmental conditions, and the eggs can be transmitted to humans through the fecal oral route. Humans acquire Toxocariasis through accidental injection of infective Toxocara species eggs from contaminated soil such as gardens, sand pits, and playgrounds, including contaminated water, contaminated raw vegetable, fruits, or utensils. Next slide, please. Now, the image you have there is the image of the life cycle of human infection with Toxocariasis. As you can see from the image, eggs are usually voided from the definitive host, that is cat or dog, into the environment, and within 2 to 6 weeks of being passed out, the eggs becomes embryonated and become infective to human, and human gets infected when it accidentally injects this infective egg from foods or contaminated water, and the eggs become hatched in the intestine, penetrate the intestinal wall, enter the blood circulation, and migrate to various tissues such as the heart, the liver, the lungs, the eye, the brain. That is it. Next slide, please. Now, there are 4 distinct clinical entities associated with human Toxocariasis. The first is visceral larval migrans, which is the most common syndrome in infected people, particularly in children, with clinical signs such as coughing, wheezing, or cutaneous manifestation. The next is ocular larval migrans, which is also more common and mostly reported in the children. And this ocular larval migrant can lead to blindness of the eye. Then covert Toxocariasis is difficult to diagnose clinically due to its non-specific nature of these symptoms. And the last one, neurotoxocariasis or cerebral toxocariasis, occurs mainly in middle-aged people, and the syndrome relates to the migration of Toxocara canis in central nervous system. Next slide, please. Now, exposure of human to Toxocara has been linked to poor school performance and cognitive deficit in children, asthma, and also blindness. Next slide, please. Now, to diagnose human Toxocariasis is often a challenge to clinicians due to the non-specific nature of the symptoms. However, diagnosis can be made using conventional methods, which include blood tests, that is blood count eosinophilia, then histopathological examination of the affected tissue if it contains the second stage larva, and this one can be diagnosed morphologically or morphometrically. Next slide, please. Other diagnostic techniques for human toxocariasis include molecular techniques such as PCR amplification to identify larval DNA in tissue or body fluids, DNA hybridization, restriction fragment length polymorphism, sequencing of toxocarial nuclear ribosomal DNA. Next slide, please. And in epidemiological studies, serological methods are used, and these include ELISA, that is enzyme link immunosorbent assays, and Western blot using Toxocariasis species secretory antigens. Now, based on the magnitude of its prevalence and its association with various diseases and clinical manifestations, Toxocariasis is a relevant disease that should have received greater attention from public health authorities. Like many other parasitic diseases, Toxocariasis is much more common in developing countries, including Nigeria. However, the disease is highly understudied in Nigeria and is poorly known because it is not a disease under epidemiological surveillance as in most African countries. Next slide, please. Now, what we want to look at are the studies that have been conducted on human Toxocariasis in Nigeria. Currently, the mean seroprevalence of Toxocaria species in general population in Nigeria is about 53%. Up to date, there are only 6 published papers on human Toxocariasis, and it comprised 5 cross-sectional studies and 1 case report. Out of these 6 studies, 4 of the studies were from the southwest region of Nigeria, while the other 2 are from the northwest and north central region of the country. Next slide, please. Now you can see the map of Nigeria showing the various locations of cities where studies on human Toxocariasis have been conducted. The first study was conducted in Jos, which is north central region of Nigeria, in year 2000. The next was in 2015 in Lagos. No, Kano State was the next, followed by Lagos in 2015. Then Ibadan in 2016 and Ile-Ife 2020. Next slide, please. Now, like I said earlier, the first study on human Toxocariasis in Nigeria was published in year 2000, and it was a cross-sectional study which comprised of pupils of Jos University Primary School, students of Jos University Demonstration Secondary School, and volunteers. The study was conducted at Jos, Plateau State, North Central Nigeria. The serological diagnosis was by a laser technique, and the seroprevalence of Toxocaria species reported was 29.8%, and the only risk factors identified in the study was geophagia. Next slide, please. After the first study, which was conducted in 2000, there was no epidemiological study on human Toxocariasis until about a decade and a half later. The second study, which was a case report, was conducted among 42 children which had retinoblastoma from the Department of Ophthalmology, Bayero University, Kano State, Northwest Nigeria. The study was conducted between 2001 and 2005. And the tissues that were collected from the children were examined histopathologically, and it was observed that Toxocariasis larvae were found in one patient given a prevalence of 2.5%. The study was conducted by Abdul and others. Next slide, please. Then the third study was conducted in 2015. This is an epidemiological study. Like I said, it was a decade and a half when the second study after the one conducted in just was conducted, and the population study comprised primary school children from Makoko, Lagos State, Southwest Nigeria, and the serum was analyzed by the use of Western blotting. The seroprevalence of Toxocariasis species reported was 86.1%. And the risk factors identified in that study included the older age, drinking of boiled water, consumption of raw vegetable, and contact with dogs. Then, following that, the fourth study on human toxocariasis in Nigeria was conducted in year 2016. And the study population comprised school children from Ibadan, Southwest Nigeria. Say that the serum analysis was by the use of ELISA technique, and the seroprevalence of Toxocariasis reported in that study was 87.5%, and it was conducted by Okewole. Next slide, yeah. The fifth study was conducted in 2017, and it was conducted by myself and other researchers. It comprised the preschool children, which was selected from 4 peri-urban communities in Osun State, Southwest Nigeria. The serum analysis was by Western blot technique, and the seroprevalence of Toxocariasis obtained in that study was 37.3%. The risk factors for the infection was identified to include age of the children, contact with dogs, and parents' religion. The last study, the sixth one, was conducted in 2020. This study was conducted by my master student whom I supervised, and the study population comprised pregnant women who attended antenatal clinic at the University Teaching Hospital, Ile-Ife, Southwest Nigeria. The serum diagnosis was by the use of Western blot technique, and the seroprevalence of Toxocariasis obtained in that study was 92.49%. Out of the 6 studies conducted, 3 of the study employed the Western blot technique, which has higher specificity compared to ELISA as the diagnostic method for detecting toxocariasis. And the use of this technique, that is the Western blot technique, was made through the collaborative study between the department, my own department of zoology, Obafemi Awolowo University, and Department of Molecular Pathology and Tropical Diseases, Taipei Medical University, Taipei, Taiwan. Next slide. Now, sources of environmental contamination with Toxocariasis species and route of transmission. The infection of human with Toxocariasis in Nigeria might have been due to the heavily contaminated environment with Toxocariasis species. The animals host responsible for this infection, that is dog and cats, sources have shown in Nigeria that they harbor high prevalences of Toxocariasis species. And the studies conducted among puppies in Nigeria, a high prevalence of 80% was reported. And for adult domestic dog, prevalent range of 33.8% to 41.7% were reported from various studies conducted on the adult domestic dogs. Also, a prevalence of 48.5% for Toxocariasis infection in cats was also reported. Next slide. In Nigeria, dogs roam about uncontrolled in public places, parks, playgrounds, and around residences, releasing their feces, which often contains hundreds of thousands of Toxocariasis into the environment. When the eggs are passed out under suitable environmental conditions, the eggs becomes embryonated and becomes infective, thereby posing a major public health risk. As it has been mentioned earlier, that humans acquire Toxocariasis through a range of routes, which includes contaminated soil, contaminated water, food items such as raw vegetables and fruits, and also contact with dogs or cats, because studies have shown that the hair of these definitive hosts harbor embryonated eggs. Yeah, one of the routes of transmission is soil, and in Nigeria, studies have examined soil contamination in various locations. And from Ibadan, Southwest region, soil examination revealed a contamination rate of 6.1% for Toxocariasis species eggs. From the federal capital, Abuja, the soil examination revealed a contamination rate of 49.2% with Toxocariasis egg. And the third one, from Samaru, Zaria, northwest Nigeria, a contamination rate of 27.2% was reported for Toxocariasis eggs. Next slide, please. Now, as I said earlier, the ears of the dog is a route by which humans can acquire Toxocariasis. And in a study conducted by Sowemimo and Anyoni in 2016, the study demonstrated the presence of Toxocariasis eggs on the ear of Nigerian dogs, which can serve as a route for acquiring infection from dogs or cats. Also, the injection of Toxocariasis eggs in raw vegetables is another route, and studies have shown from two locations, Zaria and Kaduna, from food items examined, that Toxocariasis eggs were reported from this food item in northwest region of Nigeria. Discussion. Now, from the available few published studies on human Toxocariasis in Nigeria, there is evidence that humans are frequently exposed to Toxocariasis infections. The seroprevalence value of 80% and above reported among school children from Lagos, and also the study among pregnant women from Ile-Ife, southwest Nigeria, is an evidence that exposure to Toxocariasis species is extremely common in Nigeria. Furthermore, next slide, please. You can see that high burden of Toxocariasis in Nigeria. High burden of Toxocariasis in the studies conducted in Nigeria so far may be due to prevalent favorable transmission factors such as 1, pet ownership with poor veterinary care. 2nd, poor personal hygiene. Socio-economic factors such as poor sanitation and favorable climatic conditions for the embryonation and survival of Toxocariasis eggs. So these are factors that favor high burden of Toxocariasis in Nigeria, which was seen in the studies conducted. Next slide, please. Toxocariasis, as shown in one of the studies, might also result in retinoblastoma in children, which can lead to enucleation of the eye. From the studies conducted so far, these various risk factors were identified, and they include the age, contact with dogs, drinking of unboiled water, consumption of raw vegetable, and parents' religion. Several individuals are likely to be exposed to various risk factors associated with Toxocariasis. Next slide, please. Some apparently healthy individual could be infected with Toxocariasis, and such people might be at risk of developing visceral larval migrant, ocular Toxocariasis, or covert Toxocariasis and associated complications. Therefore, there is need for public awareness campaign on Toxocariasis and associated risks in Nigeria. Studies on the seroprevalence of Toxocariasis in Nigeria are scanty. As you can see, there's only 6 studies have been conducted so far, and the increasing high seroprevalence recorded in recent studies should be of great public health concern. Now, in order to reduce the human infection with Toxocariasis, these are the possible approaches to this, and these include: 1, increasing the awareness of food hygiene. Making sure that there is education among the populace to ensure that foods that are being consumed are not contaminated with the eggs of the parasite. Also, limiting free roaming of dogs within residence and playground, because by this unrestricted movement, eggs that are deposited along with the feces into the environment, thereby contaminating the environment. So also, regular antiemetic treatment of dogs and cats so as to reduce their worm load, thereby decreasing the eggs deposited to the environment. We know that dogs and cats harbor quite a lot, hundreds of thousands of eggs. So antiemetic treatment will reduce the worm load, thereby reducing the amount of egg deposited in the environment. And other prevention measures also include washing hands after playing with pets or when exposed to potentially contaminated sites. Next slide, please. Yeah. In conclusion, there is the need to conduct more epidemiological studies on humans, animal, and environment in different geographical zones in Nigeria. especially zone not yet covered. Actually, in Nigeria, there are 6 geopolitical zones. And out of these 6 geopolitical zones, it's only 3 zones that you have a record of human toxocariasis. The other region, which includes Southeast, South-South, North-East, there are no record. So studies need to be conducted to ascertain the status of human infection with Toxocariasis. And also, further studies should be collaborative involving different disciplines, including veterinary and public health professionals under one health strategy. This will involve simultaneous investigation of humans, dogs, and the environment to better understand the epidemiology of the disease and to optimize control efforts. Thank you for listening.